Good morning. Today we are going to talk about the motility and secretory activity of the small intestine. The objectives are review the functional anatomy of the small intestine, describe the various types of movements of the small intestine, so this will cover the motility of the small intestine, describe the various sources and types of intestinal secretions, understanding their physiological relevance, and learn their neural and hormonal control. Then we'll define the term peristaltic rush and understand its physiological and pathophysiological relevance, and finally to deduce the effect of excessive loss of fluids via the small intestine during diarrhea. As you know, the small intestine is the part of the alimentary canal that lies between the stomach and the large intestine, and it has three parts: the duodenum, the jejunum, and the ileum. The small intestine is the major site for the digestion, both mechanical and chemical, and also is the major site for the absorption of food, salts, water, and orally administered drugs or medicines. It serves several functions. These include: it mixes digestive secretions with food and propel chyme along the intestine, and this is mechanical digestion. It digests and absorbs nutrients. It absorbs the constituents of the digestive secretions, the different digestive secretions: saliva, esophageal secretion, gastric secretion. After they perform their function, these secretions or the components of these secretions are not lost in the fecal material. After they perform or finish their function, they are absorbed back into the blood to be reused again. Also, the small intestine, as we knew from the previous lecture, is the major regulator of gastric function. It has an inhibitory role both neurally and hormonally. It inhibits the gastric secretion and emptying if we deliver large amount of chyme to the small intestine. It is also important for the synthesis and secretion of different hormones that influence feeding and gastric function. And finally, the small intestine defends the body against invasion by microorganisms, as we'll see that the small intestinal glands have special cells called the Paneth cells that have some defensive functions. As you learned from anatomy, the small intestine is the longest part of the alimentary canal. It has a length of 6 to 7 meters in a cadaver and 2 to 4 meters during life. It is longer in the cadaver because after death the smooth muscles in the muscularis externa lose their tone, so they will relax and make the small intestine longer than during life. As I said, it has three subdivisions, and these three subdivisions are the duodenum. It is mostly retroperitoneal structure and it has a length of 25 centimeter. The jejunum, it is intraperitoneal except for the site of attachment of the mesentery. It has a length of 2.5 meters long. And then we have the ileum, which is again intraperitoneal structure and has the length of 3.6 meters. So the longest part or subdivision of the small intestine is the ileum. The small intestine is innervated by both parasympathetic and sympathetic fibers through the vagus nerve and the thoracic splanchnic nerves. These fibers in turn will modulate the enteric nervous system, both the submucosal plexus and the myenteric plexus. So the autonomic or the both divisions of the autonomic nervous system will modulate the enteric neurons that control secretion as well as motility. The arterial supply is primarily from the superior mesenteric artery, and the veins will drain into the hepatic portal vein via the superior mesenteric vein. The small intestine has 4 layers or 4 tunics as the other parts of the alimentary canal, and these are the mucosa, submucosa, muscularis externa, and serosa. As you can see here in this illustration, these are the 4 basic layers of the small intestine. As I said, similar to the other parts of the alimentary canal. The mucosa is composed of epithelium or mucous membrane, the lamina propria and the muscularis mucosae that separate the mucosa from the submucosa. The submucosa contains the submucosal plexus plus blood vessels. The muscularis externa is composed of 2 layers, the circular muscle layer, longitudinal muscle layer, and these 2 layers are separated by the myenteric plexus, which is the main plexus that controls the motility, and as I said, it is modulated by both divisions of the autonomic nervous system, but mainly the parasympathetic via the vagus nerve. And finally we have the outer layer, which is the serosa. As you can see here that we have in certain parts of the small intestine some glands that extend into the submucosa, and we call them the Brunner's gland, and they are present in the first part of the duodenum, and also the wall is penetrated by the ducts of the accessory gland. And in the small intestine, here we have the pancreatic duct and the common bile duct. Here we go further into details about the structure of the small intestine. As I said, you can see here the 4 layers: serosa, muscularis externa, the submucosa, and the mucosa. You can see that the submucosa and mucosa are thrown into circular folds. We call them the plica circularis. If we take a closer look to the mucosa, we can see that the mucosa has finger-like projections called the villi. The villi are covered by epithelial cells. We call them the enterocytes because they are enteric epithelial cells. And the core of the villus is composed of the lamina propria that contains blood capillaries and lymphatic vessel called the lacteal, which is important for the absorption of lipids. And if we take one of the enterocytes or the absorptive cells that cover the villi or one of the villi, we can see that the apical membrane, the phospholipid bilayer of these enterocytes towards the apex of the cell, we call it the apical membrane, it is thrown into small or tiny microscopic finger-like projections. We call them the microvilli. So the surface area of the mucosa is increased by the presence of 3 structural modifications. The small intestine without these structural modifications has a surface area of 1/3 square meter, 0.33 square meter, so 1/3 of square meter. But due to the presence of those structural modification, this surface area is increased dramatically because one of the main functions of the small intestine is absorption. So we need a large absorptive surface area, and we'll see how these structural modifications will increase the surface area to large extent. These structural modifications, number 1, the plica circularis, also we call them the valves of Kerckring. They are about 1 centimeter long, and they are overgrowth of the submucosa with the covering mucosa to form circular folds. Then we have the next level of structural modification, villi. They are 1 millimeter in height. They are finger-like projections of the mucosa, and if we take 1 square millimeter of the mucosa, we can find 20 to 40 villi in that 1 square millimeter. Next, we have the microvilli. Also, we call them brush border because they resemble the brush. So they have a length of 1 micrometer. As I said, they are very tiny microscopic finger-like projections of the phospholipid bilayer of the enterocytes, the apical membrane of the enterocytes. They further enlarge the surface area of the luminal border of the enterocytes, and 1 square millimeter of the mucosa contains about 200 million microvilli. These structural modifications will increase the surface area of the mucosa from 1/3 square meter to 200 square meters, so about 600 times. As I said, this is important because the small intestine is the major site for absorption, for the final absorption of nutrients after being mechanically and chemically digested. There are histological differences between the duodenum, jejunum, and ileum, and I guess that you already studied that in anatomy. Quickly, here we can see that the Brunner's glands are present only in the duodenum, and they are absent from the jejunum and ileum. The circular folds are very marked or are more extensive in the jejunum, because the jejunum is the major site for absorption, so we need larger surface area. All of them have the crypts of Liberkin, the small intestinal glands that we'll study in one of the coming slides, and they have some differences in the shape and height of the villi. For example, in the duodenum, the villi are broad and short. In the jejunum, as it is the major absorptive site, they are long, finger-like, and in the ileum they become a bit shorter. With regard to the Paneth cells and the Peyer's patches, the lymphoid tissue, they have similar amounts. Now, the small intestine exocrine glands. The small intestine has exocrine glands, and also it has an endocrine function because it has enteroendocrine cells that we'll study in the intestinal secretions. Here you can see that this is the gut, the alimentary canal, a summary illustration showing the summary of the difference between the structure of the different parts of the alimentary canal. The esophagus, we have esophageal glands that are mucosal and submucosal. The gastric glands are mucosal only. There are no submucosal gastric glands. The esophageal glands are very few, and they release mainly mucus and bicarbonate. Here in the small intestine, this part, you can see that the duodenum has submucosal glands, and these are the Brunner's glands, and we can see mucosal glands throughout the small intestine. We call them the small intestinal glands or the crypts of Liberkin. We also find similar glands in the large intestine, but they have difference in function that we'll discover in the next lecture, inshallah. As I said, the duodenum is characterized by the presence of the duct that penetrates its wall, and this duct is the common bile duct and the pancreatic duct. I want you to notice that the difference in the shape of the villi, broad, short, long finger-like, and then short finger-like, or little bit shorter in the ileum. As you can see also in the colon, we have the crypts of Liberkin, but we don't have villi. So the surface doesn't contain villi. So the Brunner's glands are located in the submucosa of the first few centimeters of the duodenum, between the pylorus and the ampulla of water. The crypts of Liberkin, or the intestinal glands, I prefer to say small intestinal gland to differentiate them from the large intestinal glands. They are located over the entire surface of the small intestine mucosa between the base of the villi. As you can see here, the opening of the crypts of Liberkin are between the bases of the villi. As you can see in this illustration, this is a part of the small intestine wall. We can see the 4 layers: the serosa, the muscularis externa, and inner circular, outer longitudinal, separated by the myenteric plexus. Then we have the submucosa that is rich in blood vessels, lymphatics and nerve fibers, and the muscularis mucosa that separates the mucosa from the submucosa. And you can see here the mucosa is thrown into finger-like projections. These are the villi with a core of blood vessels and lymphatics. And between the villi, between the bases of the villi, we can see these small glands. These are the crypts of Liberkin, or the small intestinal glands that contain different cells, as we'll see in the coming slide. If we take a closer view to the one villus and the crypt of Liberkin, we can see that the villus is covered by absorptive cells or absorptive enterocytes that have the microvilli in their apical membrane, tiny finger-like projections of their phospholipid bilayer. In addition to that, between these absorptive cells, we can see goblet cells that secrete mucus. Again, it is rich in bicarbonate. Then, in the crypts of Liberkin, or the small intestinal glands, we can see the enteroendocrine cells that secrete different hormones, and we'll see examples of those hormones. At the base of the Paneth cell of the crypt of Liberkin, or the small intestinal gland, we can see another type of cells called the Paneth cells. These cells secrete lysozymes and also are able to perform phagocytosis. That's why the small intestine plays a role in defending the body against microorganisms. This illustration is a bit simplified. If we have a better one, we could see that there are also the stem cells, the cells that are very active in mitosis, and these mitotic cells will generate new cells that will be pushed upward to the tip of the villi, and the old cells are shed off the tip of the villi. So the small intestinal mucosa contains many deep crevices lined with glandular epithelium. Cells lining the crevices form the crypts of Liberkin, or the small intestinal glands, and secrete the intestinal juice. The crypts of Liberkin contain the following cells. Number 1, the Paneth cells that secrete lysozyme, a bactericidal enzyme, and defensins. These are small proteins with antimicrobial powers, and also Paneth cells are capable of phagocytosis. The enteroendocrine cells, it is a big family of cells. They secrete different hormones. So we have different types, and here I'm giving just a few examples. The S cells that secrete the hormone secretin, the CCK cells or the I cells, they secrete the hormone cholecystokinin or CCK, and the K cells that secrete the hormone gastric inhibitory polypeptide or GIP. As I said, not to forget the stem cells. They have high mitotic activity, with the new cells continually migrate up the villi and push off the older cells at the tips of the villi into the lumen. We have more than 10 million intestinal cells that are shed per minute, and the mucosal epithelium is replaced approximately every 3 to 5 days. So this shows us how these cells are very active in terms of mitosis. Now let's talk about the motility of the small intestine. So what are the motility patterns of the small intestine, and whether they resemble the gastric motility or not, and whether they are affected by feeding. Do we have special motility patterns during the fed state and other motility patterns during fasting, like the stomach? So the small intestinal motility, if we are in the digestive period, we have two motility patterns. These are segmentation, which is mixing contraction, and peristalsis, which is a propulsive movement. And we have another pattern that is dominant during the interdigestive period, and this is similar to the stomach. It is called the migrating or migratory motility complex. Segmentation is the primary motility pattern in the small intestine during the fed state. Please remember that the main motility pattern of the small intestine during the fed state is segmentation. It is the small intestine primary motility pattern during digestion of a meal. It mixes, as the name shows, but also slowly propels the chyme. So segmentation doesn't only mix but also propels, which is a function, it should be a function of peristalsis, but in the small intestine, segmentation is also responsible for propulsion. It consists of oscillating ring-like contractions of the circular muscles, as we see here in this illustration, along the small intestine's length. Between the contracted segments are relaxed segments or relaxed area. So there are contractions at regular intervals, and between the contractions we have relaxed area. So this relaxed area contains small amount of the chyme. The contractile rings occur at regular intervals, as I said, every few centimeters, dividing the small intestine into segments like a chain of sausages. These contractile rings do not sweep. The contractions happen and they don't move down or distally. So just they remain in their place. So they don't sweep along the length of the small intestine as peristaltic waves do, because they are not peristaltic movements. After a brief period, the contracted segments will relax and the ring-like contractions appear in previously relaxed areas. So here we have relaxed area, now they become contracted. And this process will repeat and repeat, and as you see here, this will lead to the mixing of chyme with the secretions, with the bile, with the pancreatic secretion, as well as the secretions of the crypts of Liberkin. These segmentations are initiated by the small intestine pacemaker cells, and these cells are the interstitial cells of Cajal that we had covered in the lecture of gastric motility. These cells are responsible for the basal or basic electrical rhythm. Segmentations are slight or absent between meals. There are no segmentations during the fasting state. They become vigorous or they become intense immediately after a meal. If the small intestine basal electrical rhythm brings the circular muscle cell layer to threshold, then segmentation contractions are induced, or they will appear, with the frequency of segmentation following the frequency of the basal electrical rhythm. And we studied that interstitial cells of Cajal can produce a rhythm of 12 per minute in the duodenum, and as we go distally towards the ileum, the frequency will decrease. This difference in the frequency, with higher frequency in the duodenum and less frequency in the more distal ileum, is responsible for the slow propulsion of chyme induced by segmentation. If we reverse these numbers, then the chyme will be propelled from the ileum back to the duodenum. So we need that higher frequency in the duodenum compared to the ileum, and this what makes segmentation able to propel chyme slowly up orally or towards the ileum. The excitability of the small intestine smooth muscles moving the BR closer or farther from threshold, and thus the intensity of segmentation is influenced by different factors. Number one, distension of the small intestine. We know that mechanical stimuli are very important in the alimentary canal. They are very important stimuli to induce motility, including the small intestine. Another factors that influence the excitability are hormones, example gastrin, and not to forget the extrinsic nerve activity in the form of parasympathetic and sympathetic modulation of the enteric nervous system. And we know very well that parasympathetic is the main modulator of the enteric nervous system in the alimentary canal. So the segmentation has neural control, and this neural control in the form of intrinsic nerves. Example, the gastroenteric reflex. Distension of the stomach with chyme will stimulate my enteric plexus to conduct from the stomach to the small intestine. Also, the duodenum starts to segment primarily in response to local distension caused by the presence of chyme. So the presence of food in the stomach can induce segmentation. Also, the presence of chyme in the duodenum can induce segmentation. So this is neural control mediated by mechanical stimuli or distension. Extrinsic nerves can modify the strength of these contractions. Parasympathetic stimulation enhances segmentation, whereas the sympathetic stimulation depresses segmentation or segmental activity. We have also a hormonal control of segmentation. Stimulating hormones for segmentation are gastrin, cholecystokinin, insulin, motility, and serotonin. Inhibiting hormones are secretin and glucagon. Segmentation of the empty ileum is brought about by gastrin secreted in response to the presence of chyme in the stomach, and we call this gastroileal reflex. So the presence of food or chyme in the stomach will induce segmentation neurally through the intrinsic nerves, as we saw in bullet 1a, and also brought about by gastrin that is released in response to the presence of chyme in the stomach. Now, what are the functions of segmentation? What is the benefit behind having segmental activity in the small intestine? Segmentation serves the dual function of mixing the chyme with the digestive juices secreted in the small intestine lumen and exposing all the chyme to the absorptive surface of the small intestinal mucosa. So it mixes with all the secretions, bile, pancreatic, and intestinal juice, and while mixing, it will expose this chyme to the absorptive or the huge surface area of the absorptive mucosa. Also, segmentation slowly moves chyme through the small intestine. Chyme slowly progresses forward because the frequency of segmentation declines along the length of the small intestine from 12 per minute to 8 or 9 per minute, as I said previously. The pacemaker cells in the duodenum spontaneously depolarize faster than those farther down the tract, with segmentation contractions occurring in the duodenum at a rate of 12 per minute compared to only 9 per minute in the terminal ileum. Because segmentation occurs with greater frequency in the upper part of the small intestine than in the lower part, more chyme on average is pushed forward than is pushed backward. As a result, chyme is moved slowly from the upper to the lower parts of the small intestine. Now let's cover the second motility pattern that happens during the fed state, which is peristalsis. In addition to the propulsive effect of segmentation, chyme is also propelled through the small intestine by peristaltic waves. But in the small intestine, these peristaltic waves are weak and travel for short distance compared to the stomach. These can occur in any part of the small intestine, and they move towards the anus suborally at a velocity of 0.5 to 2 centimeters per second. These peristaltic waves are faster in the proximal intestine and slower in the terminal intestine, but they are normally weak and usually die out after traveling only 3 to 5 centimeters, so they are short lasting. So forward movement of chyme is very slow, so slow that the net movement along the small intestine normally averages only 1 centimeter per minute, and this means that we need 3 to 5 hours for the passage of chyme from the pylorus to the ileocecal valve. Now let's talk about peristaltic rush, which is a special motility pattern that happens if we have intense irritation of the intestinal mucosa. Although peristalsis in the small intestine is normally weak, intense irritation of the intestinal mucosa, as occurs in some severe cases of infectious diarrhea, can cause both powerful and rapid peristalsis, called the peristaltic rush. So it is powerful and it travels a long distance, and the aim is to get rid of this strong irritant. This is initiated partially or partly by the nervous reflexes that involve the autonomic nervous system and the brainstem, and partly by intrinsic enhancement of the myenteric plexus reflexes within the gut wall. So it has both extrinsic and intrinsic neural components. The powerful peristaltic contractions travel long distances in the small intestine within minutes, sweeping the contents of the intestine into the colon, and thereby relieving the small intestine of the irritative chyme and excessive distension. So it is like a protective mechanism. It is called peristaltic rush. It is not a normal pattern of motility during the fed state. It happens only if we have intense distension or intense irritation of the mucosa of the small intestine. Now, the motility pattern that happens during the interdigestive state or the fasting state, which is called the migrating or migratory motility complex. During periods of short fasting, when most of the meal has been absorbed, the stomach and small intestine exhibit a unique motor pattern or motor activity. We already explained that in the previous lecture. The intestinal segmentation contractions will cease or stop and will be replaced by migrating motility complexes, or we call the intestinal housekeeper activity. MMC cycles that repeat every 100 minutes will sweep the intestine clean between meals, and the same for the stomach. Migrating motility complexes are regulated between meals by the hormone motilin, which is secreted during the unfed state by the endocrine cells, M cells, of the small intestine mucosa. So the M cells are responsible for the release of motilin during the fasting state or the interdigestive state. When the next meal arrives, the migrating motility complexes will stop and the motor activity associated with the meal will take over, because motilin release is inhibited by feeding. The ileocecal sphincter prevents contamination of the small intestine by colonic bacteria. The large intestine is full of bacteria, and we'll study them in the lecture of large intestine, and we'll talk about them, the colonic microbiota, and what prevents the contamination of the small intestine with colonic bacteria is the ileocecal valve that separates the last segment of the small intestine from the large colon or large intestine. Now, what are the secretions of the small intestine? The second part of the lecture. The usual question, does the intestinal juice contain any enzymes? And some students will say yes. We will see whether this is correct or not in the coming slides. The small intestine secretions. Each day, the exocrine gland cells in the small intestine mucosa, and we are talking about the crypts of Liberkin, secrete into the lumen of the small intestine about 1.5 liters of intestinal juice. It has a slightly alkaline pH, about 7.6, and this solution is composed mainly of water, salts, and mucus, and this is called succus entericus, or the juice of the intestine. The secretion increases after a meal in response to local stimulation of the small intestine mucosa by the presence of chyme. So the presence of chyme in the small intestine will trigger or stimulate the secretion from the crypts of Liberkin. The mucus in the secretion provides protection and also acts as a lubricant. The aqueous secretion provides plenty of water to participate in the enzymatic digestion of food. We need water because digestion is hydrolysis. Hydrolysis means we need the water molecule. Hydrogen on one side and OH on the other side when we break down a bond. So the aqueous secretion provides plenty of water to participate in the enzymatic digestion of food. Digestion involves hydrolysis, bond breakage by reaction with water. Please remember this bullet. No digestive enzymes are secreted into the intestinal juice or into the succus entericus. The small intestine does not synthesize and release digestive enzymes. The enzymes produced by the absorptive cells are part of the microvilli. They are membrane proteins embedded in the phospholipid bilayer that forms the microvilli. That's why we call them the brush border enzymes. So no digestive enzymes are secreted into the intestinal juice. The small intestine produces digestive enzymes that are embedded. They are not released into the lumen, embedded in the brush border membrane of the epithelial cells that line the small intestine mucosa. Now, what are the secretions by the Brunner's gland that are present in the first part of the duodenum? These glands secrete large amount of alkaline mucus in response to tactile, which means mechanical, or irritating stimuli on the duodenal mucosa, vagal stimulation, which causes increased Brunner's gland secretion concurrently with increase in stomach secretion, and gastrointestinal hormones, especially secretin, because the stimulus for S cells is the low pH in the or low or acidic chyme in the duodenum. The function of the mucus secreted by the Brunner's glands is to protect the duodenal wall from digestion by the highly acidic gastric juice that is emptied in small amount from the stomach into the duodenum. In addition, the mucus contains a large excess of bicarbonate ions, which add to the bicarbonate ions from the pancreatic secretion and bile, and these bicarbonate neutralizes the hydrochloric acid that enters the duodenum from the stomach. Brunner's glands are inhibited by sympathetic stimulation. Therefore, such stimulation in very excitable people is likely to leave the duodenal pulp unprotected and is perhaps one of the factors that cause this area of the gastrointestinal tract to be the site of peptic duodenal ulcers. Okay, so the sympathetic or intense sympathetic stimulation of the Brunner's gland in people that are under stress will decrease the Brunner's gland secretion, decreases mucus and bicarbonate formation, and will put these people at risk of a peptic ulcer in the duodenal bulb. Now, what are the secretions by the crypts of Liberkin? The surface of both crypts and the villi are covered by an epithelium composed of two types of cells. We have a moderate number of goblet cells which secrete mucus that lubricates and protects the intestinal surfaces, a large number of enterocytes which in the crypts secrete large quantities of water and electrolytes, and over the surface of adjacent villi reabsorb the water and electrolytes along with the end products of digestion. So the enterocytes in the crypts, in addition to the Paneth cells, enteroendocrine cells, and the stem cells, we have other cells that are secretory enterocytes. They secrete water and electrolytes, and this water plus the electrolytes, in addition to the products of digestion, are absorbed by the enterocytes on the villi. We call them the absorptive enterocytes. So we have two types of enterocytes: secretory enterocytes in the crypts, absorptive enterocytes on the villi. The intestinal secretions are formed by the enterocytes of the crypt and are rapidly reabsorbed by the enterocytes of the villi. So it is just like recycling water and electrolyte, and while reabsorbing the water and electrolytes, food ingredients or the simple molecules of nutrients are absorbed along them. This flow of fluid from the crypts into the villi supplies a watery vehicle for the absorption of substances from chyme when it comes in contact with the villi. As I said, this water will act as a vehicle carrying the simple molecules that resulted from the mechanical chemical digestion near the absorptive cells to be absorbed. So what is the mechanism for the secretion of this watery small intestinal gland secretion of fluid? The exact mechanism that controls the marked secretion of watery fluid by the crypts of Liberkin is still unclear. They don't know exactly the mechanism. It is believed to involve at least two active secretory processes. One is the active secretion of chloride ions into the crypts, and this also there is, in addition to that, active secretion of bicarbonate ions. The secretion of both ions causes electrical drag of positively charged sodium ions through the membrane and into the secreted fluid as well. In the stomach, we saw that the proton pump will pump hydrogen, and this hydrogen will drag chloride. Here in this crypts of Liberkin, negatively charged ions are secreted first actively into the lumen, and they will drag positively charged ions like sodium to follow them into the lumen, and both ions will drag water by osmosis. So finally, these ions together cause osmotic movement of water into the lumen of the gland and then into the lumen of the small intestine. What are the consequences of small intestinal fluid and electrolyte loss? Vomiting and subsequent loss of acidic gastric contents leads to metabolic alkalosis, and we spoke about that in the tutorial. It is what? Hypokalemic, hypochloremic metabolic acidosis, and we explained the causes of hypokalemia or the mechanism of hypokalemia. Excessive loss of intestinal contents causes dehydration, loss of nutrient or nutritional materials, and metabolic acidosis resulting from the loss of bicarbonate. And here when we talk about loss through diarrhea, not vomiting. Diarrhea is a condition characterized by the passage of highly fluid fecal material, often with increased frequency of defecation. Not only are some of the ingested materials lost, but some of the secreted materials that normally would have been reabsorbed are lost too. I said that most constituents of the digestive secretions are reabsorbed after they finish their function. But here in diarrhea, we will lose what we eat plus what the accessory and the digestive glands secrete. So we have loss of many things. Loss of fluid and acid-base imbalance. And that's why we have either alkalosis or acidosis plus dehydration. The abnormal fluidity of feces usually occurs because the small intestine is unable to absorb fluid as extensively as normal. What are the causes of diarrhea? The different causes of diarrhea are: the most common cause of diarrhea is excessive small intestinal motility. So we have less time for absorption or to complete digestion and do the absorption, which arises either from local irritation of the gut wall by bacterial or viral infection of the small intestine, or from emotional stress. Rapid transit of the small intestine content does not allow enough time for adequate absorption of fluid to occur. Diarrhea also occurs when excess osmotically active particles such as those found in lactase deficiency are present in the digestive tract lumen. These particles cause excessive fluid to enter and be retained in the lumen, thus increasing the fluidity of feces. Another cause of diarrhea are toxins of the bacterium, like the cholera toxin, the causative agent of cholera, and certain other microorganisms promote the secretion of excessive amounts of fluid by the crypts of Liberkin. So the cholera toxin will stimulate the secretory cells in the crypts of Liberkin to produce large amount of intestinal juice, and we call this secretory diarrhea. And this will result in profuse diarrhea, electrolyte imbalance, and severe dehydration. Now, I said intestinal secretion, and I stress on this concept. I said intestinal secretion contains 0 enzymes. The enzymes are part of the plasma membrane of the enterocytes, and because the microvilli resemble a brush and the enzymes are there, we call them the brush border enzymes. Brush border enzymes are digestive enzymes synthesized by intestinal absorptive cells and inserted into the plasma membrane of the microvilli, which is the apical membrane of the absorptive enterocytes. They include 4 carbohydrate digesting enzymes: dextrinase, maltase, saccharase, and lactase; 2 protein digesting enzymes called peptidases, and these aminopeptidase and dipeptidase; and 2 nucleotide digesting enzymes, and these are nucleosidases and phosphatases. And finally, these are the references from which I prepared the lecture. Thank you very much for your patience, and I welcome any questions, or if you need any clarifications, please send them by email. Thank you and have a nice day.